Today our topic is resistance to antibacterial agents. But to understand it better, first let's revise what is bacteria. Bacteria is a small single-celled microorganism that can't be seen with naked eye and generally called as germ. They play a very vital role in our atmosphere and even in our body. But despite of their importance, they also cause various diseases to living beings. To cure those diseases caused by bacteria, many medical agents are employed. These agents are called antibacterial agents or antibiotics. Antibiotics are substances that are derived from various species of microorganisms and are capable of inhibiting the growth of other microorganisms when administered in small concentrations. They can be either bactericidal, they kill microbes directly, for example, penicillins, isoniazid, or bacteriostatic, they prevent microbes from growing, for example, tetracycline, chloramphenicol. These antibacterial acts on different site of actions and follow different mechanisms to either kill or stop bacteria. The major modes of action are inhibiting cell wall synthesis, inhibiting protein synthesis, inhibiting nucleic acid synthesis, injuring the plasma membrane, inhibiting the synthesis of essential metabolites. We will briefly look at them one by one. First mechanism by which antibacterial act on microbes is by inhibiting cell wall synthesis. Peptidoglycan is major component of bacterial cell walls. Some antibiotics targets the synthesis process and consequently, the cell wall is greatly weakened and the cell undergoes lysis. Examples include penicillin, cephalosporins, carbapenems, etc. Next mechanism is by inhibiting protein synthesis. Some antibacterial inhibit bacterial protein synthesis by selectively targeting the 70S ribosome of prokaryotic cells, that is bacterial cell, while 80S ribosome of eukaryotic cells, that is host cells, remains least affected. As ribosome is directly involved in protein synthesis, so it ultimately affects the protein synthesis of bacterial cell that is a vital process for all living cells. Among the antibiotics that interfere with protein synthesis are chloramphenicol, erythromycin, streptomycin, and the tetracyclines. Next is inhibiting nucleic acid synthesis. A number of antibiotics interfere with the processes of DNA replication and transcription in microorganisms. Some inhibit DNA synthesis by targeting DNA gyrase, enzyme that helps with the winding and unwinding of circular DNA during replication in bacteria, for example, fluoroquinolones. Some antibiotics interfere with messenger RNA synthesis by targeting RNA polymerase, main enzyme involved in transcription, for example, rifampin. Next, by injuring the plasma membrane. Certain antibiotics, especially polypeptide antibiotics, bring about changes in the permeability of the plasma membrane that result in the loss of important metabolites from the microbial cell that are needed for their survival. Next mechanism is inhibiting the synthesis of essential metabolites. Some antibiotics inhibit synthesis of essential metabolites of bacterial cells that are required for their survival and growth by competitive inhibition of their enzymes with closely related substance of their substrate. For example, para-aminobenzoic acid is important substrate for the enzymatic synthesis of folic acid, but sulfanilamide acts as antimetabolite, and by binding with that specific enzyme, it leaves no active site for para-aminobenzoic acid. Thus, there'll be no folic acid synthesis in microorganism, and ultimately, no synthesis of protein and nucleic acid, leading to death of bacteria. After getting this brief knowledge about what are antibacterial agents and how they work, now let's look at the title of our topic once again, that is, resistance to antibacterial agents. Let's illustrate this concept. Human populations often have a relative resistance to diseases to which they have been exposed for many generations. For example, When Europeans first colonized tropical climes, they proved highly susceptible to diseases they had never been exposed to, although the local populations were relatively resistant. Similarly, antibiotics represent, in a sense, a disease for bacteria. When first exposed to a new antibiotic, the susceptibility of microbes tends to be high, and their mortality rate is also high. There may be only a handful of survivors from a population of billions that have developed resistance against that antibiotic. Resistance occurs when a previously susceptible organism is no longer inhibited by an antibiotic at a concentration that can be safely achieved in clinical practice. Resistance can develop quickly because bacteria multiply rapidly, or mutations arise regularly. The genes can be transferred rapidly by bacteriophages, plasmids, or other mobile genetic elements. The resistant microbes usually have some genetic characteristic that accounts for their survival, and their progeny are similarly resistant as these mutated genes transmit vertically, from generation to generation. Some genetic differences arise from random mutations. These mutational differences can be spread horizontally, that is, among same generation of bacteria as well, by transformation, conjugation, transposons, and integrins. Let's see, what are these transmission mechanisms? Starting with transformation. Many bacterial species incorporate naked DNA into their genome, a process called transformation. This naked DNA makes them resistant towards antibiotic activity, as altered gene will alter the affinity of the product. For example, Streptococcus pneumoniae and Neisseria gonorrhoeae incorporate small sections of penicillin-binding protein genes from closely related species to produce a penicillin-binding protein that binds penicillin less avidly, so becoming more resistant. Next is conjugation. Bacteria contain plasmids, circular DNA structures that are found in the cytoplasm. Many genes are carried on plasmids, including those that encode antibiotic resistance. Plasmids can pass from one bacterium to another by conjugation, allowing resistance genes to spread rapidly in populations of bacterial species that share the same environment, for example, within the intestine. Now, transposons and integrins. Transposons and integrins are mobile genetic elements able to encode transposition and move between the chromosome and plasmids, and between bacteria. Many functions, including antibiotic resistance, can be encoded on a transposon. Sensitive bacteria that get these antibiotic resistance encoded transposons thus acquire resistance. There is another concept that you must learn here is multiple resistance. Resistance against more than one type of antibacterial actions is termed as multiple resistance. Once a resistant gene is established, either by genetic alterations, random mutations, or by any other process, it can readily acquire resistance to another agent by transmission, conjugation, transposons, or integrins. Once there is more than one resistance gene, exposure to any of these agents will permit survival of the organism, which increases the risk of further resistance being acquired. Now let's move towards few major mechanisms of resistance that bacteria develop against antibiotics. It includes antibiotic degradation, impermeability, efflux mechanisms, alternative pathway, and alteration of the target site. Starting with antibiotic degradation, it can be done by enzyme inactivation. Many species of bacteria produce enzymes that destroy the structure of antibacterial and make them inactive. For example, many strains of S. aureus produce an extracellular enzyme, beta-lactamase, which can break open the penicillin beta-lactam ring, thereby inactivating it, all by enzyme addition. Some bacteria express enzymes that add an inactivating chemical group to the antibiotic, so inhibiting its activity. For example, some bacteria may become resistant to aminoglycosides by adding an acetyl, amino, or adenosine group to the antibiotic molecule. Another mechanism of resistance is impermeability. Some bacteria are naturally resistant to antibiotics, because their cell envelope is impermeable to that particular antibiotic. For example, Pseudomonas species are impermeable to some beta-lactam antibiotics. Next, efflux mechanism. Some bacteria may employ efflux mechanism for resistance against antibacterial agents by the acquisition of an inner membrane protein that actively pumps the antibiotic out of the cell, an efflux pump. Bacteria, for example E. coli or streptococci, may become resistant to tetracyclines, macrolides or fluoroquinolones. Another mechanism is alternative pathway. Bacteria may acquire genes that create an alternative pathway, other than originally required for the formation of desired product. It can circumvent the metabolic block imposed by an antibiotic. For example, S. aureus becomes resistant to methicillin or flucloxacillin when it acquires the gene MEK-A, which encodes an alternative penicillin-binding protein that is not inhibited by methicillin. Now, last mechanism for resistance is alteration of the target site. In some bacteria, resistance developed by alteration of target site of antibacterial, so their activity on target site can be blocked. For example, rifampicin acts by inhibiting the beta subunit of RNA polymerase in bacteria. RNA polymerase gene is altered by point mutations, insertions or deletions. The new RNA polymerase is not as easily inhibited by rifampicin and resistance occurs. So, this was all about the mechanisms of resistance acquired by bacteria against antibacterial actions. This video must have helped you in developing clear concept about what are antibiotics, how they act, and how bacteria show resistance to antibacterial. For more such videos, keep visiting sqadia.com.